Today I'd like to talk about our work done during my postdoc time in EMBA Heidelberg. As Monica mentioned, I did molecular and cell biology work during my PhD time and have become very interested in the dynamic process of the biological systems. And particularly now, I'm very fascinated by the beauty of the dynamic development of the embryos. But to understand such development processes, we need the approaches to monitor and visualize the dynamic processes as they take place. In a way, live imaging is one of the best ways to go beyond snapshots and to reveal the new mechanisms underlying the development. And among many different species, the mammalian early embryos are one of the most exciting models to understand their unique feature in the development, which is symmetry breaking and subsequent tissue patterning. Then our lab has extensively used mouse embryos to elucidate how the fertilized egg breaks the symmetry and how the embryos develop the pattern, establish the pattern of the inside out and outside, and also how the embryos develop into the blastocyst, consisting of 3 different lineages with the lumen inside. But basically, those events occur before the embryo implantation into the uterus. And then to monitor such pre-implantation embryonic development, we have established a right sheet live imaging system in collaboration with Ralph Funegel Lab and Jan Ehrenberg's group in EMBA. I hope you can appreciate that with this method, we can capture highly dynamic processes in the development without compromising their development, and which helps us to understand the mechanistic insights into the development. But now we are moving forward to a more complex systems of the embryos, which is the embryos beyond the implantation. So once mammalian blastocysts become mature, they implant into the uterine tissue. In the case of the mouse, this happens around embryonic day 4.5, and within 24 hours, they develop into the cylindrical morphology, which is called egg cylinder. During this morphogenesis, the mural side of the outer layer trophectoderm attaches to the uterine tissues, while the polar side of the trophectoderm develops into the extraembryonic ectoderm, EXE. Meanwhile, the neighboring epiblast tissues shown in blue here, which eventually gives rise to our body, develops into the radially aligned epithelial pattern out of cell aggregates. So despite the importance of this major morphogenesis and tissue patterning for subsequent development processes such as gastrulation, we still don't fully understand how those blastocysts develop into egg cylinders. And also we don't know what cellular dynamics underlie those epiblast patterning. But this is because our understanding so far is still quite relying on the snapshots of fixed embryos obtained by the embryo recovery from the uterus at the stages before and after implantation. Therefore, to fill the gap between pre and post-implantation embryo development, we needed to establish the ex-vivo culture system that allows us to monitor the peri-implantation development. So the left panel is the embryos at E4.5, right before the implantation. And after 6 hours, it is E4.75, the mural trophectoderm indeed show the invasive protrusion to adhere the uterine tissues. And then after 1 day, so E5.5, 5.5, the polar trophectoderm already fully developed into the EXE. Then together with our lab members, Huiting and Laura, we attempted to develop a culture system covering this period of the peri-implantation development by using a 3D gel containing matrigel and collagen to support the embryo development in 3D. Basically, this idea is coming from that post-implantation embryo is supported by the uterine soft puffy tissues. But however, it failed to support proper embryonic development in 24 hours. So specifically, polar trifectum maintained a single cell layer, as you can see here, and then they didn't develop into the extramembrane ectoderm. And also, at the earlier time point, those polar trifectum cell shapes are remarkably different between the cultured embryo and the in utero developed embryos. So those polar trifectum get columnar shape after 6 hours from before the implantation, but those trifectum are still stretched in the cultured condition. Then to know what is happening during this culture system, we live image the embryo development in 3D by using inverted light field microscope, which is Invisbeam from Luxembourg. Now, by using two objectives, one is for illumination, the other for detection, we can we can monitor the development in 3D. And as you can see here, the inside tissues containing epiblast mass undergo the proliferation, but the outer layer of the trifectum are still in the way the single layer, and they don't form the EXE. So therefore, the embedding whole embryos in 3D gel broke the egg cylinder morphogenesis, and we needed to investigate further to know why it didn't work. Then we carefully looked into the polar trifectum cells by doing immunostaining against actin and also dephosphorylated myosin regulatory light chain. It revealed that the actin as well as phosphorylated myosin are strongly accumulated at the apical cortex in the in utero developed embryos, while they are only localized at the cell-cell junction in the cultured embryos. So this suggests that the higher cortical tension is applied to the outer trifectum cell layer, and then stretching them and then maintaining them in a single cell layer. And furthermore, this observation was confirmed by the direct measurement using micropipette aspiration, and showing that the cortical tension indeed increased twofold over the 6 hours of the culture. Therefore, too much higher cortical tension seems to prevent the polar trifectum from invagination. And then we wondered if the tension release would enable the polar trifectum constriction and also the invagination. To test this hypothesis, we employed laser ablation to cut the cell-cell junction in order to release the junctional tension between the mural trifectum